Hello, everybody. Here we are again with a new topic that is forensic science. Forensic science and genetic fingerprinting. Of course, specifically, in this section, we shall talk about genetic fingerprinting. But we start from definition of forensic science. Therefore, this is one part of application of the knowledge of biology at technological level to help humanity. Then first, let us ask, what is forensic science? What is forensic science? This is simple definition for forensic science. Forensic science refers to the use of science to help solve legal cases or criminal acts, or the use of science to law and criminal investigations. It's simply helping humanity in terms of legal cases or criminal acts. Then, uh, what is the significance of the contribution of the knowledge of biology, the science of biology in its modern context? Biology, of course, generally speaking, biology holds huge applications in forensic science through many disciplines, of course. For example, just blood typing is one way we can solve criminal cases or legal cases in humanity. The other is fingerprinting, the general fingerprint. They're just the fingerprints, which are, of course, fingerprints are different even between identical twins. Identical twins who share the same genetic makeup. Fingerprints are different in identical twins. This also helps. Of course, it is not the essence of this section. Of course, the other is forensic entomology. Forensic entomology. Forensic, just legal case, solving legal cases or criminal acts by using the science of entomology. Entomology is the study of insects. Of course, um, it's very young. We are going when the criminal case involves days. The first organism that visits the dead body is the flies. Flies, different types of flies, different species of flies visit the dead body. They feed some secretions and they lay their eggs there. The eggs are incubated, they undergo metamorphosis, worms or larvae are formed. Therefore, we are studying this part of the insects by the entomology. We are applying the incubation period, the process of change of egg into larva, the larva, pupa, whatever it is. Therefore, we can at least estimate the time of days by taking samples of insect insect eggs and their changes in the dead body. This is part of what is known as forensic entomology. Therefore, this also helps in solving criminal cases and legal cases in humanity. The fourth, the topic that is the focus of this portion is genetic fingerprinting, also known as DNA fingerprinting. Of course, it is also known as molecular fingerprinting. If you like, it has also a fourth name that is DNA profiling. Therefore, our focus is in this part, in this section, in this part that is genetic fingerprinting. Otherwise, as I've tried to tell you earlier, the science of biology holds huge application in forensic science through blood typing. Not only the usual AB, and O type of blood, and there are many blood genetics like Rh factor analysis, MN blood typing, whatever. There are almost about 11 genetic typing of the human blood. The most common that we know is the AB or blood grouping system. This also blood typing helps us to solve legal cases or criminal acts, of course. And fingerprinting, Especially the blood typing is used in solving paternity cases, of course. Fingerprinting, the fingerprints are taken, fingerprints to identify who the individual is, just identity markers, because I've told you that these fingerprints are different even in between identical twins with the same genetic makeup. Forensic entomology, this is a science of entomology, applying the science of entomology or insect study, insect knowledge on our insects, especially in terms of their reproduction, that is metamorphosis, so that we can investigate criminal cases that involve death of a victim. And the focus of our part, the red part, you see, genetic fingerprinting. What does it mean? We are adding genetics to the fingerprint. It's not the usual traditional fingerprint. It is the fingerprint of our genes, gene profile. I have told you there is one secret by which we all are different. We all are different in less than 1% of our DNA. This less than 1% of DNA, highly variable part of DNA, which is non-coding or which is not producing proteins, are the basis of genetic fingerprinting. We cut out this from sample cells taken from our body, then we do some sort of techniques, molecular techniques like gel electrophoresis, then we identify individuals, because the non-coding less than 1% part of our DNA are highly variable among humanity. Otherwise, humanity shares more than 99% of the DNA similar, identical or similar, of course. Therefore, we shall talk about this issue later on in this section. Now you can see the general, the usual traditional fingerprint is the middle one, fingerprint. We call it simply fingerprint. Whereas if we identify the specific DNA profile, that is DNA fingerprinting or genetic fingerprinting or molecular fingerprinting or DNA profiling or molecular profiling, whatever you call it. Genetic fingerprinting, the other names as I have told you, DNA fingerprinting, DNA profiling, molecular fingerprinting, all these are under this section. Let us ask, what is genetic fingerprinting now? We have tried to define what forensic science, just using science to solve the human problem in terms of criminal cases or legal cases. Now let us focus on genetic fingerprinting or DNA fingerprinting or molecular fingerprinting or DNA profiling. All these are same names for the same issue. This simple definition. Genetic fingerprinting is a technique used to identify individuals based on their unique DNA patterns. I have told you what makes that unique DNA pattern. Those less than 1% of the human genome, less than 1% of the human genome, which are highly variable among humanity around the world, they are the basis. Okay, we will come to the point. Therefore, simply genetic fingerprinting can be defined as genetic fingerprinting, a technique used to identify individuals based on their unique DNA patterns. It allows DNA samples from different people to be compared to look for similarities and differences. This comparison, we do profile for individual suspects in case of criminal case. You may have your suspects. You compare the DNA profile of the suspects and you take sample from the crime scene. If there is bleeding while the while the criminal is doing his killing act or his rape act, if he has left some mark, his own DNA mark there, we take that DNA, we do profile from the suspects, we take sample and do profile. If these profiles from the crime scene match with the profile from one of the suspect, or more than one can do the same crime, more than one can do the same crime. If they match, that is we can be sure that the suspect is the real doer of the criminal act. Therefore, genetic fingerprinting allows DNA sample from different people to be compared to look for similarities or differences. And forensic biologists inspect crime scans. They go and inspect, they study the crime scans, take samples from the crime scan. You see, they inspect crime scans to examine potential sources of evidence using samples of DNA from, say, from blood extracted dry or wet blood. If it is just recently made crime, of course, you may get wet blood before it dries. Even you can take scrap from the dried out blood in the crime scene. Just blood is one very important source of cells from which we get extract the DNA, those DNA which are unique in individuals. The second is buccal or cheek cells. Cheek cells, easy to obtain cheek cells. If you scrap the inside of cheek, you can take out lots of cells easily without causing damage or bleeding. Cheek buccal, or we call it cheek cells, are taken from the inner surface of our cheeks. And the other is semen. Seminal fluid is also one source of that DNA. It could be dried, can be analyzed years later. If you can dry it and it can be analyzed years later, you can preserve it if you like. Hair roots, you pull out hair, the root of the hair contains cells. These cells, root hair cells, root follicle cells, are important source of DNA also. You can take. Even you can take saliva. From saliva, you can get cells and DNA from, so those cells can be used for DNA profiling. Skin graft, skin cells, you can take some graft of skin cells, especially. Of course, it's painful to take some grafts of skin cell without anesthesia from living human. But you can take simply skin cell grafts from dead bodies. Just a small sample of cell is just taken from the dead body without, of course, pain. Therefore, this is known as skin graft. These are all sources of the DNA that we require, because these all contain cells. Inside cells in the nucleus, they do contain chromosomes, and the chromosomes are the seeds of the DNA that from which we extract those pieces of DNA that we require. I will come to the steps how genetic fingerprinting is made. Therefore, these are forensic biologists inspect crime scans to examine potential sources of evidence using samples of DNA from all this blood, buccal sample, cheek cells, semen, hair roots, saliva, skin cells, skin grafts. All this can be the sources of cells from which DNA is extracted. Forensic biologists then help to solve crime cases through analysis of physical evidences found at the crime scene and on victim of the crime, and compare it with evidences related to the suspect. This is what forensic biologists are doing, generally speaking. Forensic biologists, not only forensic geneticists, all generally speaking, forensic biologists, whatever. That biologist could be blood biologist or insect biologist or genetic biologist. Generally speaking, forensic biologists help to solve crime cases through analysis of physical evidences found at the crime scene and on victim of the crime, and compare it with evidence related to the suspect. Specifically, if that forensic biologist is forensic genetist, what he or she does is take samples of cells from blood, buccal smear or buccal sample, or from skin graft, semen, whatever, and take out DNA and cut and isolate those needed DNA, which are highly polymorphic, highly different among individuals, and do what? DNA profiling. Just focus, the focus is by doing DNA profiling, the forensic genetist helps to solve legal issues or criminal acts. Also help confirm if people are related to each other, like in paternity testing. The forensic biologists also solve the problem of paternity issues, paternity testing, because the mother is always the mother. Mother is always mother to her born children. Okay? But we don't know who put that sperm. We don't know. Especially if the mother is not loyal to her husband, mistakes can be made. Therefore, that is paternity testing. Is that man the real father of this boy or girl? That is one legal case that brings people to the law courts. Therefore, forensic geneticists or forensic biologists are called to solve this problem, paternity testing. And if you like, they are also called upon to testify in court, give evidence in legal cases where the forensic biologists are asked to come based on their analysis and their findings to give their evidence, to be witness to the legal case. This technique was developed, this forensic science, this technique was genetic fingerprinting. This technique, the genetic fingerprinting, was developed in 1984 by British genetist Alex Jeffrey, most controversial, one of the most controversial scientists with many dark sides, of course, Alex Jeffreys, who discovered that certain sequence of human DNA is highly variable. Earlier, I've tried to tell you what is the basis of genetic fingerprinting, this highly variable part of human DNA, which are known as mini satellites. Mini satellites, of course, they are named by many different names. I will tell you the names: mini satellites, or variable number tandem repeats, or short tandem repeats, whatever. These many satellites are repeated within genes and vary significantly among individuals. You see, this is the basis for genetic fingerprinting. Highly polymorphic. Highly polymorphic means highly variable. They are different between you and me and him, her, whatever it is. Therefore, if we cut out this and do genetic profile by following the procedure, the DNA profiles between the chance that two DNA profiles to be identical between two individuals is very, very low. Mini satellites are the basis. Therefore, Alec Jeffrey first identified that while he was studying the human DNA, he identified that certain regions of the DNA are highly variable, and they are known as mini satellites. And these are repeated within genes and vary significantly among individuals. Repeated sequences of nucleotides that are highly variable among individuals. And let me add some note here. It is a reality that the DNAs of two randomly taken individuals are more than 99% similar, and they differ only by less than 1%. I have tried to tell you earlier. You see, this is what we are. More than 99% of our DNAs are similar. In fact, some put this number as 99.9% of our DNA. 99.9% of our DNA is similar. Only we differ by 0.1 when they exaggerated, of course. Some put it like that. And here I put it like this. Of course, similar, but in different ways. More than 99% of our DNA is similar, and we differ from each other only by less than 1% of our DNA. That is, that less than 1% of DNA is responsible for the basis for genetic fingerprinting, which allowed genetic fingerprinting to be conducted and be used in legal case or criminal acts or paternity testing. Then, and it is this small difference that is the base of genetic fingerprinting. The small difference is the base of what do you call this small difference? The short tandem repeats, or the variable number tandem repeats, or mini satellites. And it is this small difference that is the base of genetic fingerprinting. And this less than 1% difference is in the non-coding region of the DNA. Non-coding region, by the way, non-coding region of DNA means, generally speaking, the part of the DNA whose instruction is not used for protein synthesis. Coding part of DNA is transcribed and translated so that protein is synthesized. Non-coding region of DNA is not directly used for protein synthesis by transcription and translation. Therefore, it's not coded. Its instruction is not coded into messenger RNA. Therefore, these mini satellites are non-coding regions of the DNA. Of course, in fact, this mystery also. Do you know this? The fact that about a maximum of 2% of our DNA is coding. Only about 2% of our DNAs are the coding. Imagine, what about the remaining 98%? Non-coding. Non-coding. Only 2% of our DNA is used for protein synthesis through gene expression, that is transcription and translation. These are the two fundamental processes of the central dogma of molecular, central dogma of what we call this gene expression, central dogma of molecular biology, gene expression. Therefore, these mini satellites belong to the 98%, that is non-coding region of the DNA. But what makes them quite different from the rest is they are repetitive, yes. They are highly polymorphic, that means they are different between X and Y and Z individuals. And that's why they are being used as sources of genetic fingerprinting. And this non-coding region shows great polymorphism. I have to read. What is polymorphism? Variation, simply variation. Variation among the world population, among people. Variation among people. They show high variability, great variability, and such variable regions of our DNA are repetitive, heterogeneous, repetitive. The other property of these microsatellites are repetitive, heterogeneous. That means they differ among stars. Classes of nucleotides within or between genes. They are found between genes, scattered between genes, within genes. These 10 to 100 nucleotides long, repetitive heterogeneous microsatellites are, generally speaking, 10 to 100 nucleotides long, and with 2 to 10 base pairs repeats. They are repeating units, like this repetitive heterogeneous class of nucleotides are just like they are repeating. If it is with 4 nucleotides, for example, AAGC, this repeats AAGC, AAGC, AAGC. It repeats 3 times in one individual. If the same sequence is there in another individual, it may repeat 5, 6, 7 times, whatever, 10 times repeating. You see, therefore, with 2 to 10 base pair repeats that show high variability among individuals. Therefore, remember, they are highly polymorphic, they are repetitive nucleotide sequences, and they are non-coding. Their instruction is not used for protein synthesis. And these non-coding repetitive sequence of nucleotides are named as follows. They do have many names: short tandem repeats, STRs, mini satellites, or variable number tandem repeats, if you like. Don't forget this, very important, crucial. If you are asked, what made genetic fingerprinting possible? Oh, please, the presence of these short tandem repeats, the presence of mini satellites or variable number tandem repeats. Everybody has this non-coding sequences. What is the special feature of this sequence of nucleotides? Oh, they are um, repeating units. This repetition is the same, not the same in individuals. They may repeat from 2 to 10 nucleotides. For example, if it's 2, AC, 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 how many times? It could repeat 10 times or 15 times or 7 times or 6 times or 4 times. Depends. Therefore, they repeat. Starting from 2 nucleotides, it could be 3, 4 nucleotides long. Each repeat, it could be 2 nucleotide long minimum and maximum 10 nucleotides long. I will show you. I do have example here. Therefore, don't forget, the non-coding repetitive sequence of nucleotides are named as short tandem repeats, mini satellites, or variable number tandem repeats. This is my example. Look, this personality with the man in the glass at the top. Say AG80 repeats, AG80, AG80, AG. How many times does it repeat this personality? It has AG80, AG7 repeats, you see, at one part of the DNA. At another part of the DNA, he has another AG80 repeat, AG80 repeat, 6 repeats. At the same position, the personality in the middle, a woman or a girl, the same repeat, she has the same region, AG80, AG80, 4 repeats. You see variation? At the same region, corresponding region to the first personality, she has 19 repeats, AG80, AG80, AG80. And the last, the man, red beard and red hair and personality, the bottom, the same locus or the same place on the chromosome, which is labeled as STR, short tandem repeats, site number 1, short tandem repeat, site number 2. And this short tandem repeat, site number 1, the same sequence, AG80, this personality has 12 repeats. And in site, short tandem repeat, site 2, AG80 repeats, 14 repeats. You see the variation? This is the essence. This is what allowed genetic fingerprinting. If you cut out these pieces of DNA and run electrophoresis, I will tell you about electrophoresis, how we cut out these pieces of DNA from different individuals and search for similarity or differences in the process known as gene electrophoresis, so that we can do genetic profile. The profile of Mr. X and Mr. Y totally different. I will show you. Then application. The variable number tandem repeats or short tandem repeats allows for the creation of a unique DNA profile for each person, and with the exception of identical twins. Of course, identical twins, though they vary by the usual fingerprint, they are different in their tandem repeats or the genetic profile. The VNTRs can be cut out by restriction enzymes. Specific restriction enzymes are used. They are amplified by PCR, of course, amplification. And there is also one section that I teach you about what PCR, polymerase chain reaction. You have to know polymerase chain reaction. You take sample cells from the sample cell, you take DNA, and from the DNA, you use restriction enzymes and separate those short tandem repeats. If the separated short tandem repeats are not enough in amount to run your Gel electrophoresis, you multiply them. Amplification means simply cloning the genes or multiplying them. How do we do this? Of course, in vitro. That means outside of cells. That means in a machine, in a lab, test tube machine. We put those fragments and we multiply them so that we will have enough of those samples of broken samples of a DNA to run the gel electrophores. Therefore, simply amplified by amplification or um, cloning, also known as cloning the genes, the cutout restriction, the cutout short tandem repeats are multiplied to have much amount of sample of each repeat for to run the gel electrophoresis, if you like, once or two times or three times. You can repeat it. Therefore, polymerase chain reaction is artificially multiplying these pieces of genes isolated from the DNA. And then after amplification, of course, we do another procedure. I've told you, electrophoresis, or it is also known as gel, G-E-L, gel electrophoresis. Also, I will show this. Therefore, first you use restriction enzymes and cut out and isolate. Then you multiply this cut out short tandem repeats or the piece of DNA by using polymerase chain reaction in a special machine known as thermal cycler. Then you run gel electrophoresis to see the DNA profile or genetic profile of individuals. As individuals, we likely have different numbers of repeats at a given satellite DNA satellite DNA locus or place. They will generate unique DNA profiles. You see, as individuals, will likely have different number of repeats at a given satellite DNA locus or at a given short tandem repeat satellite short tandem repeat extraction locus or place on the DNA. They will generate unique DNA analysis, profile, and longer repeats will generate larger fragments, while shorter repeats will generate smaller fragments. So you know that, yes. And when examining the variability of the satellite DNA sequences, the odds that two random human individuals, would share the same genetic pattern by chance depends on the number of loci. From how many different places did you take this short tandem repeat? One loci, two loci, one locus, two loci, three loci, four loci. It depends. Therefore, as the number of short tandem repeats taken from as the number of loci we take the satellite DNA increases, the chance that two individuals will have the same DNA profile significantly drops or decreases. Therefore, example, if we take the VNTRs from just one locus and do DNA profiling, the chances are two random individuals having the same DNA profile is 1 in 10. It's not 1 in 10. Highly likely that the two DNA profiles will be similar. 1 in 10 the chance. Again, we are increasing the loci. And if we take the VNTRs from two loci, the chance of having the same sequence is 1 in 100. Now you see we are decreasing the chance of two individuals having the same DNA profile. Again, if we take the VNTRs from 10 loci, the chance of having the same sequence is 1 in 10 billion. Oh, significant decrease in obtaining different DNA profile. You see, this is what we need. Now, I'm going to show you what the CIA is using. The CIA, the Central Intelligence Agency in America. CIA is in its genetic lab usually uses not 10, 13 it takes to be sure. What will happen if we take the VNTRs from just 30 loci? I'm sorry, this is a problem. But he read it just like this. If we take the VNTRs from 30 loci, cancel just one loci, 13 loci, this is what the CIA does. The chance of having the same sequence is 1 in 10 trillion. 1 in 10 trillion. Trillion is 10 raised to 12. Almost no chance for two individuals having the same DNA profile. So you can be sure if this suspect is the real criminal. You can tell with 100%, almost 100%, 100% just uh, uh, sure that you tell this personality is the crime, this suspect is the one who did the criminal act. Therefore, sorry for this problem here. Cancel just one locus, and if we take the VNTRs from 13 loci, the chance of having the same sequence is 1 in 10 trillion. I'm telling you, this is the number of loci by which the CIA is doing to investigate criminals or suspects for his own research or for its own mission. And therefore, the number of loci from which random repeats are taken increases, we can minimize the chance of two individuals having the same or similar DNA profile, so that we can be sure this Mr. X is the innocent and Mr. Y is the criminal. We can say that with almost 100%. Being 100% sure. And now, how it works, let us see how it works. DNA profiling or genetic fingerprinting. The process of genetic fingerprinting involves several key steps. What are these key steps? The first, sample collection. You collect sample blood, sample semen, sample skin grafts, whatever. DNA can be extracted from various biological samples such as blood, hair, skin, saliva. Then DNA extraction. The DNA is isolated and purified from the collected cells. Then isolating the repeating fragments from the DNA. Then from the DNA, we take out those short tandem repeats or the VNTRs, very variable number tandem repeats from the DNA sample by using the appropriate specific restriction enzymes which cut the DNA into specific points. Then fragmentation. The DNA is cut into fragments using restriction enzymes which target specific sequences within the DNA. Then gene cloning or amplification. I've told you we use the machine thermal cycler. Gene amplification, the isolated cut out short tandem repeats or the VNTRs are multiplied to have larger sample if we like to do the experiment or the gel electrophoresis. Many times we need large amount of sample so that we multiply the isolated pieces of short tandem repeats or VNTRs. Since the sample fragment obtained from the suspects of the crime scene are not enough to do the DNA profile, the sample DNA fragments are multiplied in a machine called thermal cycler in a process known as PCR, polymerase chain reaction. And this process is gene cloning. You are multiplying, simply multiplying genes artificial, or it is also known as in vitro gene cloning. In vitro means in artificial media in the lab, in a machine test tube. The genes are put in test tube, they are put in thermal cycler, special device here, can show you. Then we run electrophoresis. After we obtained enough sample, we run electrophoresis, a procedure. I will show you also. The fragments are separated based on their size using a technique called gel electrophoresis. And gel electrophoresis is a separation of biomolecules, of course, DNA, RNA, protein. You can separate. In this case, DNA profiling is done, DNA is separated based on their size, the size of the broken pieces or the fragments, and charge. DNA has a negative charge, allowing for analysis and interpretation of the results. You see, when we run gel electrophoresis, the tray that we use for gel electrophoresis is connected to electric source. It has one pole, anode, and the other pole, cathode, negative, positive poles. We put the sample DNA, and the DNA fragments or DNA generally is negatively charged because of its phosphate. So negative is attracted by positives, therefore the DNA migrates towards the positive anode, so that this migration depends on the size. Those with smaller size migrate faster towards the anode. Those with heavier size move slowly through the gel substance, agarose or agarose gel they call it. I will show you. This migration produces bands of this DNA profile, which can be taken, image can be taken from these bands of fragments of DNA, and DNA profiling is done. I will show you the whole process. Therefore, we do an electrophoresis after amplification. This is what gel electrophoresis is done. How it's done? You see, this is glass plate. The first one is glass plate or casting tray, they call it. It's made of glass. This casting tray, we boil and sterilize and pour a solution, liquid agarose solution, agarose, on this glass plate. It will solidify. The agarose is porous. Inside there are many pores. It's not dense if we use the appropriate amount of agarose and boil. Therefore, it solidifies in the third tray. You see, solidify, and we bring comb. They call it comb in order to dig a hole. You see, the third tray. The comb is pushed down so that it produces the holes that inside which we put the sample DNAs, the wells, WELLs, wells. For example, you can see here 1, 2, 3, 4 wells. Therefore, we put all this, the samples from different individuals are put in this well. Then this glass plate is attached to anode, electric source, power source, anode cathode, and the DNA migrate towards the anode because they are negatively charged, and you know it, opposite charges attract each other. So DNA is negatively charged and it is attracted towards the anode, the positive side. Then when they migrate, the migration is in such a way that smaller size fragments move faster and larger fragments move very slowly, and this brings about the difference in the DNA profiling. You see, the rule is that it says DNA is negatively charged and the fragments move towards a positively charged anode, and shorter or smaller fragments move faster through the gel, creating a distinct pattern. Then we are taking this, of course, each X-ray photograph is taken and analysis made. The resulting DNA pattern is analyzed and compared to other samples to establish identity or relationships. It could be paternal relationships or identity of personality, so that we can decide this person is criminal and this person is innocent. Therefore, this is how it is done. For example, The first one, she is a victim being killed by somebody. We don't know who is that somebody. Could be suspect one, suspect two, suspect three. And we have obtained sample from the crime scene, the place where uh, the crime has been made. We have obtained a knife there. When the person trying to cut and kill the woman, he might have cut his own body while she was struggling. He might have. Therefore, you may obtain blood of the criminal, either on the knife or on her shirt or body, whatever. If he has raped her, you can get sample from her vagina, of course, seminal. Therefore, you do profile of the crime scene, the victim, in case she might have killed herself, in case. Therefore, we need also the DNA profile of the victim, DNA profile of the crime scene, and DNA profile of suspects under control, or maybe may not be under control. Some may flee, of course. Then, if you get a match between one of the suspect and the crime scene, therefore that suspect is the responsible for the crime. You can see here. You can see individual two is with the red, long red hair and beard, mustache also. The second suspect number two is, according to this genetic profile, suspect number two is has similar DNA profile with the crime scene genetic profile. Therefore, we can be dead sure that suspect 1 and 3 are just released. They are free of the crime, innocent. Suspect number 2 is responsible for this crime. Damn, killer, dirty personality.